Hello, I'm Kostas Spirgakis, and I am the Dissemination and Exploitation Manager of the Secreted project. And today we have the honor to have Dr. Jose Jimenez from Imperial College London to enlighten us about emerging biotechnology and its applications in context of Secreted, in which Imperial College is also a partner.
Hi, Kostas. Thank you very much. I'm also very happy to be here, and it's a pleasure to have this opportunity to discuss our work, especially what we do in the context of security.
Thank you. Thank you very much, Dr. Jimenez. And before we deepen in the activities of Secreted, could you please give us some information about your research team, its expertise, and of course, recent advanced achievements that you could share with us?
Yeah, of course. So my group specializes in a field that is called synthetic biology. And what we do is to basically use methods, and these methods include genetics, molecular biology, mathematical models, bioinformatics, and we try to engineer different biological systems. We mainly work with bacteria. More recently, we've started working with mammalian cells as well. In all our projects, we try to improve our understanding of fundamental principles of these organisms that we are studying, but we also aim to develop applications that may have an impact in society. So, for example, we are working on a project in which we are trying to engineer organisms so that they can feed on plastic waste. And the idea is to use plastics as pretty much bacterial food and transform it into other molecules of value, for example, bioplastics, which are plastics that are biodegradable, or other molecules with pharmacological properties, like, for example, bioacid.
Thank you, Dr. Jimenez. Thank you. It's quite clear that Imperial College holds powerful tools, knowledge, and infrastructure to build capacities in engineering strains, focusing on industry-driven compounds with tailor-made properties, which is also the main goal of Secreted. And speaking about that, let me ask you about the activities and the vision of Secreted from your own point of view.
Sure. So in Secreted, we have two main roles. So the first of them is the cloning and the recombinant expression of new pathways that are being identified by other partners in the project. So we will receive DNA sequences from them, and we will use a robotic platform to clone all the genes that are required for the production of the molecules that constitute the targets of the project. All these assembled pathways will be then sent to other partners, and we will work together in their improvement if needed. The second task is the optimization of the organisms that we use for expression of all those pathways. And we have ample experience investigating bacterial physiology. And the idea here is to take advantage of directed evolution and computational methods and modeling to try to engineer the organisms that we use to produce the molecules of value so that we can increase the yields of all these different processes.
Okay, so under these conditions, let me further ask you how the activities of Imperial College are aligned with the goals and the expected achievements of Secreted.
Yeah, so these two activities are important for the pipeline of Secreted. So on one hand, the genetic designs that we are coming up with will be tested and validated by other colleagues, which will recommend the modifications that could be implemented in different iterations of a potential cycle. And on the other hand, we will also provide our partners with improved strains for the biosynthesis of the target molecules, which then need to be tested at a much larger scale, mimicking industrial processes.
Okay, so speaking about engineered strains, I have a simple question that many people have. So why do we actually need engineered strains? Why not directly use those that they already exist in nature as they are?
Yeah, this is a very interesting question that I'm afraid doesn't have a very simple answer. So these natural strains may simply not be the best ones for production. So sometimes we don't know how to culture or propagate the organisms, which could also have very poor performance when we test them in production settings, which are very specific. So other times, the organisms could maybe be pathogenic, so they could be harmful for humans, or they may require conditions that are not compatible with the process of production. So, for example, in Secreted, we work with marine organisms. They tend to be adapted to high salt concentrations. These high salt might corrode steel tanks used for production. So both in lab and industrial settings alike, we tend to work with a handful of model organisms that we can easily genetically modify, given that we need to introduce new pathways or make them more efficient. So environmental organisms in this regard are normally not domesticated, and they tend to be not suitable for this purpose.
Okay, so now the approach now seems much clearer to me, but I may also imagine that part of our audience is possible to have some prejudice about using engineered strains, namely, for instance, genetic modified organisms, if I'm not wrong, in order to produce compounds that we are using in our everyday life. So, which is the answer about these beliefs? Is it safe? Can we or our children, can we consume, for instance, food supplements that they contain bio-based surfactants or siderophores from engineered microorganisms?
Yes, absolutely. So even though it is true that we use genetically modified organisms for production, the products themselves are no different from those that we identify in nature. In fact, they're identical. So this is the key point, something that I need to emphasize and make very clear. So the product, so it could be, for example, a food supplement, it's identical to the one that we would obtain from natural sources. In fact, even from a legal perspective, in most countries, which include all the countries in the European Union, the products are not considered genetically modified once they are purified and removed from the engineered strain. Okay, so this is, for example, what happens when we make insulin. So what we do is to take advantage of the technology to improve the production so that we can obtain, for example, higher yields of the desired molecules and trying to reduce their cost, as well as trying to improve the sustainability of the process. But the products themselves are not GMOs, they are not considered GMOs, and they are safe. They're identical to the natural counterpart.
Thank you very much, Dr. Jimenez, for answering all our questions and making clear that we consume the same compounds as always, only that we investigate new ways to produce them that are more efficient and safer for the environment. So before closing, I would like to inform all people watching this Q&A that you can learn more about Secreted through its official website. You can follow and like our social media, as well as you can also subscribe in our YouTube channel and our newsletter by using the links provided below this video. Dr. Jimenez?
Yeah, thank you very much, Costas, and thanks everyone for listening. I would simply like to add that if anyone is interested in knowing more about the research that we do in biotechnology, in particular for a circular economy, which also includes, for example, the removal of plastic pollution, please have a look at the web pages that you have on the screen. We are also present in social media, and we'd be happy to answer any questions you may have in case you wanted to get in touch.
Thank you. So closing this very interesting Q&A interview, I would like to thank you again, Dr. Jimenez, for helping us understand the key ingredients and goals of emerging biotechnology. And I would also like to thank all the viewers of this video. Bye-bye.
Bye, thank you.